I hope everybody in the hall appreciate the lunch. So now it's time to talk about in silico 3D structure prediction of Aptamers. This is my work performed at the University of Technology of Compiègne in the enzyme and cell engineering lab. Work supervised by the Dr. Irene Mafucci and the Professor Aval Bihan-Perangère. Right. Okay. So as you may know, the current technique aimed to select aptamers is the SELEX process. I will not detail it, but you probably know also that despite the fact that it's one of the most advanced techniques to select high affinity aptamers, the yield remains really low, mostly because of the use of a random oligonucleotides library. And the last decade saw, it's not working anymore. Okay, thank you. So the last decade saw the development of bioinformatic tools applied to aptamers modeling, which opened the way to the use of in silico approaches as an alternative to SELEX. So next, please. So this, no. Before, please. Oh, okay. So it will allow us to rationalize the selections from the SELEX process. Next, please. So to in silico design aptamers, the aim is to first predict the structures. Next. Starting from the sequence, the aim is to first predict the secondary structure predictions in order to guide the 3D structure prediction just after. Multiple selection tools are available in either in both prediction for 3D and for secondary structure, and as they use different algorithms, the accuracy may change, and so we perform next, please, the benchmarking steps for both secondary and tertiary structure prediction in order to select the best tools and have a reliable and accurate procedure. Next. So to develop this procedure, we extracted aptamers data and oligonucleotides data from the well-known dataset and database, nucleic acid database and protein data map. Next, please. This results in a selection of all the structure which contain oligonucleotides, and it results in 12,000 entries. Next, we selected the data which contains only DNA oligonucleotides, which represent around 7,500 structure, and from this amount of data, only 177 structure are single-stranded oligonucleotides. And so this will be the data we will use for the next steps of the project. And so we store the data in two different data sets. The first one, which contains DNA oligonucleotides, which the structure have been resolved alone, so NMR structure. And also we store the data with oligonucleotides in complex with protein, which represent this amount of data. And it will be used for the benchmarking steps. Next. The first step is to predict the secondary structure of aptamers, which is basically, as you should know, the base pairing disposition, which is involved in the structure folding. This is the mainly used representation for the secondary structure. But in computational biology, we use the dot bracket notation, which is easier to handle informatically. Next. So we have dots, which means that nucleotide associate is not pair, while the open and closed brackets mean that nucleotide associate form a base pair. Starting from a sequence, so secondary structure prediction software is able to recover the most probable base pairing and to find finally the secondary structure, which is given in dot bracket format that is usable for many purposes. And here we will use it for the 3D structure prediction just after, and also, but first, the comparison of secondary structure between them. So there are different approaches to calculate the best folding of by using secondary structure prediction tools. And here it's a non-exhaustive list of the different tools available for secondary structure predictions, classified by approach. Here, thermodynamics prediction, statistical modeling, and some approaches are able to combine both approaches. Next, all those tools have been used to predict the secondary structure of Aptamers extracted before from the data selection. And we have different tools which present different algorithms, and so it may result in differences in prediction. For example, with this sequence, if we use M-fold, linear fold, MC-fold, or RNA fold, we don't have the same secondary structure. And how to know which one is the best to predict the secondary structure? We have to perform the comparison with the experimental structure extracted from the data selection first. And to do this, we use a new matrix called Aptamers that we developed, and which is more sensitive than the current use techniques, like, for example, RNA distance. Next. So I will talk about Aptamers process. Indeed, it works with the translation of the dot bracket notation into matrix of dots, where dots mean that nucleotide in X and Y coordinate form a base pair. Then we calculate the Manhattan distance in order to extract the lowest distance between the different points. Next. And so we calculate then the sum of all Manhattan distance between points divided by the number of points in both matrices to get the Aptamers distance. As it's really complicated, the algorithm, you just have to know that to consider that structure are close, we have to get an Aptamers distance value below 1.5. And so we conclude about the secondary structure prediction benchmarking by calculating the Aptamers distance for all these tools. And finally, we can see that M-fold is the best to predict the secondary structure compared to the other one, with 69% of correctly predicted secondary structure in the first data set, which contain oligonucleotide only. And for the second one, 85% structure correctly predicted for oligonucleotide in complex. So we will use in M-fold in our protocol to predict the 3D structure. Next, for the 3D structure prediction, we decided to develop a workflow and to predict the 3D structure. And also take into account the flexibility by using molecular dynamics. So here, to predict the 3D structure prediction, we need a structure of departure, which takes as input sequence and secondary structure to enforce the prediction to adopt the correct folding. And then we make the prediction with Rosetta, as it's a well-known suite of modeling software, which include features in 3D structure prediction. However, the problem with Rosetta and 3D structure prediction is that it gives only one conformation, and it cannot allow us to explore all the mobility of Aptamers structures. So we perform molecular dynamics, which is a technique based on classical physics, which allow to explore the different conformation of molecules along the time. But also molecular dynamics has some problem. Even we can explore some conformations, it's often failed to explore all the possible conformation. So, we perform some accelerated molecular dynamics. Next, yes, next please. We perform accelerated molecular dynamics, which is almost the same technique that molecular dynamics, but it's allowed to explore more conformation than classical molecular dynamics. The principle is similar, but contrary to molecular dynamics, where you can find some structure trapped in a local energetic minimum, the thing is we have to put some energy in the system in order to reach the local, to overcome the energetic barriers and reach other conformations. Here, for example, this structure was trapped in this local energetic minimum, and it reached it thanks to an energetic boost. So we have a complete protocol for the 3D prediction, and we applied it on two different structures to see what is going on. So the first structure we tried and we make the prediction on is the structure available on PDB with the code 1EZN. So initially we predict the 3D structure with Rosetta. And as you can see, the prediction, the colors are not really well, but as you can see, the prediction in orange does not really match with the experimental one. And also to see that, we can calculate the root mean square deviation, which is basically the measure of the average atomic deviations between a molecule and a reference. And so here you can see that we have a value of 13, but we expect value around 5 to conclude about structural proximity. So this is really high. And this justified next the molecular dynamic steps. Even though molecular dynamics seems to improve a bit the RMSD value and the fitting between the prediction and the experimental one, it's still really high with 10 angstrom. And as I told you, we expect value around 5. And it's still quite stable during the simulation time. So we can suppose that we are trapped in a local energetic minimum. As I told you, it's a problem. And so to explore more conformation, we applied AMD, so accelerated molecular dynamics. We applied it on both experimental structure extracted from PDB and the prediction we made with Rosetta. As it gives us a lot of conformation for both experimental and prediction, we perform a clustering to get only the most probable conformations along dynamics. And we have two populations, and we calculate their MSD, and we align them. As you can see, for these populations, we have a better match than for this one, with our MSD value of 4 for this first population. And for the second one, we are even able to reach a MSD below 3. So these are really good results. And so we can say that this protocol is pretty good to predict the 3D structure and explore conformation. We have another example to present. For this example, 5-HTO, also available in the PDB, we applied a Rosetta prediction. Again, as you can see, as the previous one, it doesn't fit with the experimental structure in red, and the MSD is really high. So we perform molecular dynamics. The same thing happens. We observe a slight improvement of the MSD, but it's still quite high. And so we perform AMD just after. But for this one, we didn't get a really good RMSD. We didn't see a really high improvement of the RMSD value as compared to molecular dynamics. But it can be explained by this. Actually, the experimental molecule and the prediction are different by this part of the molecule, the structure, which seems to rotate compared to the experimental one. And so this can explain the RMSD, which is quite high. And if we remove this part from the RMSD calculation, which involves the nucleotides 6 to the nucleotides number 15, we have a better fitting and also a good RMSD value of 2.8 again, which is really good. But this means that we have only 70% of the structure, which is greatly aligned with the experimental one. So, but this is quite good actually, because 70% of the structure is correctly predicted. But this means that we have to improve our procedure in order to avoid this kind of situation and solve this problem. So in order to be able to predict correctly an experimental, or at least reach one structure. Next. So I come to the conclusion. What about this work? So we perform a benchmarking of the secondary prediction tools, and we conclude that we have one secondary structure prediction tools, which is really good, and it's M-fold. And to do so, we develop a new method, Aptamers, which is better than others to compare the secondary structure. And this allows us to conclude about the best secondary structure prediction tool, M-fold. And so we also developed a workflow and to predict the 3D structure of Aptamers, and which considers the flexibility thanks to molecular dynamics. And so we can resume it by this. We start from a nucleic acid sequence. We predict the secondary structure with M-fold. we have, so the sequence is the secondary structure as input for Rosetta and the first 3D prediction. Then we applied molecular dynamics to explore all the possible conformation of an aptamers and oligonucleotides. No, before, please, I didn't finish. So for the 3D structure, as the 3D structure is really important for interaction, it's really good to know that we are able to predict the 3D structure. And so with a known sequence and a target, we are able to investigate interaction using, for example, other in silico techniques like docking. And using this procedure, we will be able not to replace selects, but to help selects for the selections of high affinity aptamers. Yes. So I would like to thank the International Society on Aptamers to giving me the opportunity to present today. I would also like to thank my supervisors and also the Dr. Miren Davila Felipe for participation to develop Aptamat, and also all the members of my team. And also thank you for your attention.